Here we go with mostly looking at the differences, but also the similarities between mitosis and meiosis. In meiosis, we have, first of all, replication happening in S phase before what we call meiosis even starts. So that's just like in idea, that's just like mitosis. Replication happens first. But then we have two consecutive cell divisions, not just one cell division. Two cell divisions, two cytokinesis, two meiosis, meiosis 1 and meiosis 2. But there's not a replication in between those two cell divisions, only one replication. And so that's going to result in 4 total cells, one big difference. Meiosis results in 4 daughter cells from the one you started with. Did I say that wrong? Mitosis, I forget what I said. Mitosis results in 2 daughter cells. Meiosis results in 4 daughter cells from the one cell you start with. And again, what these 4 daughter cells have is quite different than what the parent cell had. They only have one set, one of each kind. So now, this always confuses people at first. After describing what I've described, especially when we're talking about humans with 46, when you look at the simplest case you can have in a sexually reproducing organism is that in its cells, this organism only has 2 chromosomes, just 2. They're homologous to each other. So how many sets of chromosomes are in this cell? 2. What's the definition of a set? One of each kind. This cell has 2 of that kind. This is a cell down here that's got just 1 of each kind of what this cell has 2 of. So that's why this is a diploid cell. It's got 2 sets. For an organism with cells like this, what is a set? A set is just 1 chromosome. Here the word set's very confusing. There is no other set in life that you call a set that has just 1 in it, right? A set of silverware doesn't just have 1 fork. A set of golf clubs doesn't just have 1 club. But in this case, for an organism whose diploid number, the number of chromosomes in 2 sets, is just 2, a set is just 1. Now, it's unlikely that you see this used as an example, but you see it right here out of a textbook, so it's possible, okay? But now that we've gone past that, let's go back and look at this a little more closely. Before this cell, which is in G1 phase of its life, it's got single-stranded chromosomes. Before it undergoes meiosis, which is going to start here, it is going to go through S phase, and each of these will copy itself and become double-stranded, each of them. So now look what we have. We've got this set of genes and this set of genes. We got 2 sets of genes here. I shouldn't have used the word set. Okay, we've got 2 bunches of genes here. Now we have 4 bunches of genes. Where are they going to end up? They're going to end up in 4 different cells. This cell's going to have a bunch. This cell's going to have a bunch. This cell a bunch. This cell a bunch. The cell we started with had 2 bunches. The cells we end up with have 1 bunch. But you can see how we got 4 bunches to end up with 1 bunch in each of 4 cells. That's what replication made happen. So now to get from a cell that's got 4 bunches to cells that have just 1 bunch, we need 2 divisions. The first division, as you can see, is not going to pull the chromatids apart. It's just going to separate the homologous chromosomes from each other. Then a cytokinesis makes 2 separate cells, and this cell has just 1 chromosome in it. It's double-stranded right now, but we call it just 1 chromosome. That means this cell is haploid. It's just got 1 set. In this case, 1 set is just 1, right? And our second division is going to get us down to cells that have just single-stranded chromosomes. You can kind of see maybe that this second meiosis division is just like mitosis. this double-stranded thing is going to have the two chromatids pulled apart. We won't call them chromatids anymore. We'll call them single-stranded chromosomes. But this second division of meiosis is very much identical to mitosis in all the mechanics of it. But here's the key difference going back to meiosis 1. In meiosis 1, we're going to see a metaphase where chromosomes line up down the middle, but here they're going to line up with their partner. They're not going to line up in single file. They're going to line up with their homologous partner. So now we'll look at a cell. Oh, we need to get that off the screen. We're going to look at a cell now. Boom. We're in prophase. We can't see any chromosomes all coiled up, right? Now we can. What's the diploid number of this cell? Well, this cell has homologous pairs, right? It's got a pair of big ones, it's got a pair of little ones. That means this cell has two sets of chromosomes. Now they colored them for you to see that they have kind of a blue set, a big blue one and a big little one. What's the set? One of each kind, one big one, one little one. And they have a red set, one big one, one little one. Now this is what happens in prophase 1 of meiosis that doesn't happen in mitosis. In meiosis, these homologous partners find each other. And they get very close. They even literally hug each other, right? And sometimes they do something that we're going to not talk about too much now, but sometimes they switch pieces. You see it right here. Okay? We're going to ignore these words, but we'll say this word. Tetra in Greek means four, right? This is what somebody chose, slightly confusingly, to call a pair of homologous chromosomes at this stage of a cell's life, prophase 1, when they're right together. The process by which they come together and find each other is called synapsis, a Greek word that means joining. So by the process of synapsis, tetrads form in a cell. If this was a cell of yours and mine, how many tetrads would there be? 23. How many pairs of chromosomes do you have? 23. How many chromosomes are in a set? One of each of 23 pairs. Right? So far so good. What's the diploid number of this cell? 4. How many chromosomes are in two sets in this cell? 4. The 4 that you see right here. Okay? Ready? So now, here's what I said about metaphase 1 of meiosis and how different it is than metaphase of mitosis, where these 4 chromosomes would be lined up, but they'd be lined up single file, right? Like this. We'd still have 2 little ones and 2 big ones. There would be homologous pairs in this cell, but they wouldn't be paired up like they are here in meiosis 1. And this pairing is what sets the stage for the separation of 2 sets together into one set here and one set here. And eventually this is going to divide down the middle and we'll make 2 cells that each have just one set of double-stranded chromosomes. So this is anaphase 1, right? Anaphase is when things are being pulled apart. In anaphase of mitosis, the chromatids were pulled apart. The double-stranded chromosomes were pulled apart. But here, the homologs, the homologous chromosomes, are going to get pulled apart. So here we have telophase 1. Here again, a cleavage furrow clearly tells you this is telophase. We got a little nuclear membrane forming around the now 2 nuclei, but these 2 nuclei are just going to have one set of chromosomes in them. So then when cytokinesis occurs, and this continues and separates these two, we'll have 2 cells, each haploid. One set of chromosomes, right? Oops, haploid. The haploid number here, how many chromosomes are in one set for this cell? 2. If this was a human cell, there'd be 23 in there, right? One of each size, one of each kind, right? And so here we have now 2 cells that are going to divide again in the second division, very mitosis-like. Now the chromosomes are going to line up single file down the middle, metaphase 1. And, excuse me, this is metaphase 2. This is the second division, right? Anaphase 2, chromatids being separated. This is just, again, like mitosis. Then we have this happening in both cells, right? Then we have telophase 2 in 2 different cells. And then when they separate, skipping that, we have cells that have one of each kind. Okay. So here's again a list of the similarities and differences. Most of the differences occur in number, really. Mitosis produces 2 cells. Meiosis produces 4, right? The number of chromosomes in those cells after mitosis is done is the same as the number that you started with. In meiosis, it's half of the number that you started with. Here you have them side by side in cells that have 4 chromosomes, just like the ones we got finished looking at. Here you clearly see the biggest difference is how those chromosomes line up the first time they line up, which is meiosis 1, metaphase 1, compared to metaphase of mitosis, right? And then in the second cell division, they don't show all the phases here, obviously. You simply have chromatids separated. So 4 cells from the one you start with, 2 cells from the one you start with in mitosis. Same number of chromosomes as each other and the cell you start with, and half the number of chromosomes. I'm going to add here, if we started this diagram with the cell before it copied its chromosomes, there would be 4, 2 big ones and 2 little ones, single-stranded ones. Then S phase happens and they're double-stranded, right? Here would be the same. You'd have 4 single-stranded ones. S phase happens, 4 double-stranded ones. Now you're ready to do mitosis or meiosis after S phase. There we go. So I'll do these practice ones in class again. And that is mitosis.